delighted to have the opportunity to introduce my friend and my colleague and my collaborator, Jules, who I have known for what turns out to be, I think, 11 years. And so Jules and I met in, I believe, the summer of 2013, when we were both fairly new postdocs, and we took the Woods Hole course in fungal pathogenesis together and got to spend 2 weeks at Woods Hole learning all kinds of things about fungal pathogenesis and fungal mycology. And when I met her then, I thought, wow, this is a really smart, special scientist. And she has continued to make me believe that that first impression was absolutely correct. So Jules is currently leading a junior group in the departments of Mycology and Genomes and Genetics at the Institut Pasteur in France. Her group studies how fungal pathogens acquire drug resistance and how population heterogeneity can be leveraged to escape antifungal treatment. They are also interested in developing new antifungal strategies against Candida species. Previously, she was a postdoc and a research assistant professor at Brown University in the United States, and received her PhD in molecular biology from the University of Aberdeen in the UK, which is a global hotspot for fungal research excellence, for anyone who doesn't know. She's also currently a CIFAR Global Scholar in the Fungal Kingdom Threats and Opportunities program. So I'm delighted that she agreed to give this talk today, and I'm looking forward to hearing about what she's been up to lately.
Cool. Thanks so much, Eliza, for the nice introduction. And can everyone see my slides okay? Okay. So yeah, I'm happy to be here and to meet the fellows of EvoFunPath. And before I start, I just wanted to let everyone know that feel free to interrupt me if you have questions, because sometimes it can feel more organic, and don't be shy to stop me, because I'm happy to answer questions during my talk. As you know, fungal infections among human infectious diseases have been widely ignored and continue to be so. We got a recent bump last year by several species being included in the WHO fungal priority pathogen list. And among these, you'll actually find some Candida species, some of which we actually study and we're interested in the lab. You'll see that cordyceps is not on this, so the species that was featured in the Last of Us show. So these pathogens cause over 1.5 billion infections. So this is a very high number, similar to the deaths, and about 1 million deaths, over 1 million deaths per year. So this is similar to the deaths caused by TB and malaria. So we should pay more attention to them, and I'm glad you guys have a network studying them. So we are particularly interested in Candida species. So these are pathobionts. So they exist as commensal organisms in our bodies, in our genital or gastrourinary tract, on our skin. But they can disseminate and become opportunistic pathogens when our immune systems are disrupted, or when we take antibiotics, or when we have other serious conditions like diabetes or other infections. So because of this, they actually lead to these life-threatening infections, and they can have very high mortality rates. So unfortunately, we have very few antifungal drugs available, so very few classes, nothing like the classes of drugs that we have for bacteria, with azoles being the more prescribed, due to their wide spectrum of activity and their availability. Unfortunately, we have drug-resistant Candida isolates for literally all existing antifungals. And we have some species that are intrinsically resistant to some drugs, as well as species that are multi-drug resistant. So typically, most infections are caused by Candida albicans. So this remains the most prevalent Candida species. However, the resistance levels have tended to stay relatively constant over time. So about 5 to 7% of Candida albicans isolates that cause bloodstream infections are drug resistant to at least one antifungal. But recently we've seen many species that are starting to increase in prevalence, and unfortunately some of them are actually intrinsically resistant to drugs. So one example is Candida glabrata or Candida krusei. And in the last couple of years, we've actually seen a lot of outbreaks caused by drug-resistant Candida parapsilosis isolates. So these other species are worth paying attention to and monitoring more closely. We also have, as I've said, multi-drug resistant species, and I guess the poster child for these kinds of, among Candida species is Candida auris, which was first isolated from the environment in 2009. And then it kind of spread all over the globe. And the CDC used to have this map showing it spread around the globe, but this map is no longer updated given how widespread the species is. So it spread very rapidly and it's incredibly dangerous. So a few key facts about the species: it's frequently misidentified, it causes localized outbreaks. The species is very hard to clean from hospitals. Typically, people have to burn the unit or use some very extreme measures to get rid of it. It also transmits through person-to-person events. So you will also find it colonizing a lot of healthy people, including, for example, caretakers in elderly care facilities. It's highly lethal, reaching 60% in susceptible groups. More than 90% of the strains are resistant to azoles, more than 30% resistant to amphotericin, and literally you only have the echinocandins as being the drugs of last resort against these infections. So a lot of this high resistance is correlated to mutations in drug resistance genes, as well as a higher number of drug transporters and other virulence genes in the genomes of these isolates. So it's a very interesting species, but also very dangerous.
Jules, does it get misidentified as albicans?
Yeah.
Entirely.
Yeah.
Yeah.
I know.
Thanks.
Yeah, so in addition to this, we have things that kind of break the norm. So we have this paradigm that susceptible isolates should be cleared by antifungal treatment. So if you have an infection with the susceptible isolate, single round of therapy should be able to clear this infection. But this is not the reality. And in fact, we have a lot of cases with susceptible isolates that are treated with multiple rounds of antifungals, and the treatments are unable to clear these infections. So this leads to persistent infections. So we've seen that, for example, some of these infections are associated with high tolerance levels in these isolates. But overall, it's important to take home this message that actually a lot of fatal infections are not caused by drug-resistant isolates. So it means that there's a lot more out there that we need to understand about these isolates. So we think part of it is due to the fact that current classifications actually do not reflect the wide isolate diversity and the diversity of drug responses in these Candida isolates. So typically what the clinicians tend to do, and I think this is our kind of binary thinking system, we tend to classify isolates as being susceptible or resistant. And what I'm showing here are examples of disk diffusion assays where we plate a lawn of cells on a petri dish, and then we place a drug disc in the middle, and then we look at how big the area of inhibition around the drug disc is. So a typical susceptible isolate will develop this nice area, zone of inhibition next to the disc, whereas a resistant isolate, whereas for a resistant isolate, the cells will tend to grow all the way to the edge of the disc, so appearing completely uninhibited. This is an example for fluconazole, but you will see similar things for other drugs. And again, these are quite extreme examples because this isolate is super resistant. But in reality, what you see when you take isolates from the clinic and you evaluate their drug responses, you see a lot more in between. So a lot more gray area phenotypes. So for example, you can see small colonies that appear to grow more slowly in the area of inhibition. And you can see that it's not the entire population that we've plated. So it's a subpopulation that tends to do this, as well as colonies that appear to grow completely uninhibited by the drug, and they grow all the way to the edge of the disc. So this is an area of interest in our lab. We're really trying to understand how some of this population heterogeneity enables these diverse drug responses, and potentially how these other phenomena might actually enable or precede the acquisition of true drug resistance, or what we understand as drug resistance. So what antifungal drugs do we have? So we have 4 main classes of antifungals to treat Candida species. These include azoles, with the poster child fluconazole, which inhibits ergosterol biosynthesis. Polines, an example is amphotericin B, which forms membrane pores, causing ion leakage and cell death. Pyrimidine analogs, an example being 5-flucytosine, which inhibits DNA and RNA synthesis. This is not very highly used, as well as echinocandins, an example being caspofungin, which inhibits beta-glucan synthesis in the cell wall. So overall, the major themes are inhibition of cell membrane and cell wall. It's important to take note of the fact that there are drug-resistant strains for all the antifungals that we have. So we need to do better and increase stewardship. So how do these work? This is a simple schematic showing how they work in broad terms. Azoles inhibit ERG11, which is an ergosterol biosynthetic gene. Polines inhibit ergosterol in the plasma membrane directly. Echinocandins inhibit FKS1, which is an enzyme contributing to beta-glucan synthesis. And pyrimidine analogs, that said, they inhibit DNA and RNA synthesis. We have a new drug on the market. So this one came out a couple of years ago, which has the same mode of action as an echinocandins by inhibiting FKS1. Unfortunately, coming up with new antifungals is much harder in fungi than in bacteria, because fungi are eukaryotes, so many proteins that are potential targets in fungi are also found in humans. So we really need to come up with very, very specific molecules that target fungi and avoid human targets. So this is just a timeline of systemic antifungals and their discovery, just to kind of show how dire the situation is. So most recent antifungals, in addition to some of the new ones that came out last year, were developed 20 years ago. So these include the echinocandins. The ones that are still most frequently used were developed 40 years ago, whereas the one that is typically the last resort antifungal was developed 60 years ago, so amphotericin B. This one is still very potent against a lot of fungi, but it's highly toxic to humans. So best to avoid it if possible. So as I said, many fungi are intrinsically drug resistant. And unfortunately, many of these antifungals, especially azoles, are used in agriculture, which increases the number of resistant isolates that develop in the environment. A clear example of this are aspergillus isolates. And while the discovery pipeline has actually increased in the last decade, it takes a very long time to develop a new antifungal. And part of this is due to a chronic lack of investment in this area. So are we making antifungals? So there's hope on the horizon, because we have several new antifungals in late stage development. A couple of them are molecules that have new modes of action. So these are the most exciting, but we also have molecules that kind of, they seem to be improving current drugs by targeting the same mechanism, but they have like special features. So for example, these two are also beta-glucan synthase inhibitors, similar to echinocandins. This one has a new mode of action. It inhibits GWT1, which participates in synthesis of GPI anchors. So this is an important feature of fungal cell walls. This one is an azole, which is actually optimized for inhalation. And we also have another new mode of action with olorofim, which inhibits an enzyme that participates in pyrimidine biosynthesis. So it's kind of exciting, but it will be good to see some of these new drugs actually in the clinic. So how hard is it to make new drugs? So unfortunately, it can be very hard, and it can take a very long time. So it can take 5 to 7 years from first human trials to a new approved drug. It can be expensive. It can cost hundreds of millions and millions of dollars. For example, the recently developed drug olorofilm, which was developed by F2G, it costs over $200 million to develop this drug. And it took 23 years from the discovery of the initial compound to phase 2 trials. So this is a ridiculous amount of time and money, unfortunately. And I feel like we don't have this much time for development of new drugs. So in addition to these hurdles, we also have a couple of other issues. So some of the fungi that are actually emerging and are becoming quite scary tend to be geographically restricted. So the fact that they're geographically restricted means that it's less likely for them to become interesting for investors and for pharmaceutical companies. And also less frequent species are more likely to be ignored by drug developers. So things that are not very widespread, things that only show up in a certain type of year or in a particular area, they tend to be more ignored due to a potential smaller market size. So bottom line is we need novel antifungal strategies and increased stewardship for the antifungals that we currently have. So how do we combat antifungal drug resistance? So if you break it down and think about it, we as scientists have a few different options, right? So first, I think it's important to understand how Candida species escape drugs, so how they adapt and avoid antifungal exposure. But also we can work towards identifying new targets. And this, as I said, can be very hard for fungi because they're eukaryotes. But we can also work to identify molecules that can improve the lifespan of existing drugs by potentiating them, for example. So this might also have the advantage that it might block the emergence of resistance against some of these drugs that we're potentiating. So we were particularly interested in potentiating azole drugs because they are so broad spectrum and so widely used and so accessible by in the clinic because of their low cost. And if we do a literature search, we actually find that there are quite a lot of molecules out there that can potentiate or work well with azoles to clear fungal infections. So I've colored these in 3 different colors. So because these are natural compounds, repurposed drugs, or entirely new molecules. And you can see that several trends emerged as to how they act. So for example, we have a lot of them that inhibit CDR1 or MDR1, which are main drug transporters that pump out azole drugs. We have some that directly act on the membrane. So basically by disrupting membrane integrity and kind of helping azoles in that way. We have some that target mitochondrial function and therefore lead to increased ROS production. We have some that target pathways that are important for resisting azole drugs, such as HSP90 or calcineurin. We have drugs that directly inhibit drug resistance genes, for example, drug transporters or transcription factors that upregulate drug resistance genes, as well as some drugs that target the machinery of membrane synthesis. So basically target ER, Golgi, or lipid transport in the cell. So more broad spectrum, as well as drugs that directly target ergosterol synthesis, similar to what azoles do. So there's a lot of potential there, but I think there's the problem is that many of these studies are still in their infancy. We're missing a lot of studies as to would these drugs actually work in animal models of infection, or what is their exact mechanism of action. And none of these, unfortunately, have moved from academic studies to actually being used in the clinic. So there's still we still have a lot of work to do in this area. So we were particularly interested in a study that came out a few years ago that described that inhibition of vesicular transport could actually impact susceptibility to fluconazole. So this study came from Patrick Van Dijk's group, and they showed that by deleting vesicular transport gene, you can actually restore azole susceptibility. And they used this molecule here, which is called sortin-2, which is a vesicular transport inhibitor. And they saw that it actually synergized quite well with fluconazole and it was able to restore susceptibility in azole-resistant drugs. So we thought this was pretty cool because first, it seemed that like there's a lot of potential. These pathways are actually still unknown, but there's a lot of mechanisms that contribute to vesicular transport. So potentially you could have a lot of targets in this pathway that could be hit by a new drug. So at this point, we got in touch to a group that actually developed some molecules for mammalian cells. So they had a library of vesicular trafficking inhibitors that they used for to inhibit other types of infections. So we took their in-house library of over 100 analogs of mammalian vesicular trafficking inhibitors, and we asked, well, could these work against Candida species? Could we use them in conjunction with fluconazole to see would we be able to restore azole susceptibility? So the idea for our screen was, well, we'll take a resistant isolate that looks like this when we do a disc assay with fluconazole. But if we add the potentiator, hopefully we'll get something that looks like this. So we would now restore azole susceptibility. And we were lucky enough to find that actually several molecules were able to have this effect. So what I'm showing here are two examples of two isolates that are, one of them is actually super resistant and one of them is both resistant and tolerant. And we saw that in combination with some of these molecules, we were able to restore susceptibility as well as to clear tolerance for these isolates. So you can see the zone of inhibition were quite big for the majority of these compounds. So these were overall very isolate-specific effects. So in some isolates, we would see a bigger effect than in others, but overall, we try to go with the ones that seemed more potent, and again, that had an effect on both resistance and tolerance. So we were surprised to see that actually our top molecules contained 1,4-benzodiazepine scaffold. So this scaffold is actually present in a lot of drugs used to treat a variety of different conditions. You're probably more used to them being to the scaffold being present in drugs such as diazepam or lorazepam. So this is not the only type of activity that this core can have. So they're actually used as anti-cancer drugs or anti-infective. So it's a very, very flexible core, and it's continuously being explored in drug diversity and drug discovery. So basically, we were quite intrigued to see that they all had this core and just had slight variations on their moieties. So coming back to the biology of how these drugs work, we first wanted to assess what is the type of drug interaction that these molecules had with fluconazole. And to do this, we use the checkerboard assay. So this is an assay where you modulate both drugs on an XY fashion. So for example, here you have increasing concentration of fluconazole, whereas on the y-axis, you have increasing concentrations of potentiator, in our case, 1,4-benzodiazepines. And then you grow Candida cells with these different concentrations of drug to see what's the optimal combination of drug concentrations that would lead to the best outcome, so most inhibition. And there's a formula you can apply to calculate a fractional inhibitory concentration index that essentially tells you how synergistic the interaction between the two drugs is. So we calculated these Fichi scores, that's what they're called. And the scale is something along these lines. If the score is bigger than 4, it means you have an antagonistic interaction. If it's between 1 to 4, then you have indifference between the two drugs. You have an additive effect if it's between 0.5 and 1, or you have synergy if the score is less than 0.5. And these are examples of scores for 3 isolates and for 7 compounds. And what you can see is that in our case, for most compounds and for most isolates, we tended to have synergistic interactions between the drugs. So this was quite encouraging. You'll see, for example, that for isolate C, we actually have higher levels of blue, and this is because this strain was super resistant, so it had a higher dynamic range for both molecules to act. So the change in resistance was much greater. So overall, we found synergy between the potentiators and fluconazole. So you can also plot these on in a different way. So you can ask, how does susceptibility to fluconazole change according to concentration of potentiator? And this is an example of an isolate that was very resistant. So the MIC 50 started at over 128 microgram per mil. You can see that's the MIC at 0 concentration potentiator. With DMSO, nothing changes. So you get a straight line, DMSO being the negative control and the solvent for our compounds. But with increasing concentration of these potentiators, we saw significant decreases in MIC 50 levels. So for example, with this green one here, we saw that by using 12.5 micromolar of the potentiator, we were able to reduce the MIC 50 more than 100-fold. So this told us again that these molecules are actually quite good at restoring susceptibility in our Candida strains.
Can I ask a question about this?
Yeah.
What are the error bars different like replicates of the
Yes.
experiment? And so you see a lot of variation sort of in that transition between full growth and
yeah. Because it's probably not a straight line. It's probably a more dynamic process. And yeah, there's a big drop here between this one and this one.
Yeah, and I guess I just always wonder if like the amount of variation we see in our lab is just like that's just biology. Do you have any idea what causes that variation?
Probably it's, temperature outside that day, humidity, all of these things that are so hard to measure in a lab. Yeah, I mean, we have a lot of issues with these kinds of parameters that it's impossible to measure or to track on a day-to-day basis. Yeah.
Thanks. I think it's good to sort of normalize the variation in our experiments because often when we're in our lab alone, we're like, what am I doing wrong?
I know, yeah.
For presenting that, I appreciate
or have
it.
my strains changed or have my molecules changed? Yeah. Yeah, that's why it's good to do different experiments in different days so you can trust your data more. So another type of assay that we do in the lab, we do these time kill curves to basically establish if the drug combination has a static or cidal effect. And again, here the picture was not as black and white as we would have hoped to get. The assay is you take your Candida albicans cells, then you incubate them with either negative control, fluconazole, or fluconazole plus compound. And then every few hours, you take an aliquot of your culture and you plate it on a YPD plate and then count how many CFUs you get. It's a very annoying experiment to do, but it's the standard accepted method in the field to kind of address this question. Do you have a static or do you have a cidal effect? So what I'm showing here are these CFUs levels over time. So starting with 100%, so you start with 100%, and then what happens with your culture over time. So with DMSO, as expected, you get an increase in CFU numbers. With fluconazole alone, this was a fairly resistant strain. Again, we saw a significant increase in CFU numbers. So fluconazole didn't really have an effect in this strain, but with the combination, you can see that you get all sorts of responses. So some molecules being better than others. So in some cases, we got a static effect. So our cells were no longer growing. They stayed at the same level. But in some, we actually got a cidal effect with the viability of the culture going down significantly, so more than 100-fold. And again, this result was very dependent on both the molecule that we were testing and the strain that we were testing. So there's a lot of complexity behind answers like these. And it's hard to say whether something is purely static or purely cidal, because a lot of time it can be strain dependent or genetic background dependent. So we were also curious whether these molecules would work in other fungal species. So we gathered some isolates that represented species close to Candida albicans as well as further apart. So things like Candida auris or cerevisiae, but things like dubliniensis or tropicalis as well. And what I'm showing here is the heat map of how the MIC 90 levels changed in all of these assays. So the top row shows whether the respective strain was fluconazole resistant or not. And the color shows, again, if it's blue, it means susceptibility went down. So it means the combination worked. If it's orange, it means we got an antagonistic effect. So the combination did not work. So what we were surprised to see was that actually we were getting a lot more blue in species that were closer related to Candida albicans, so such as Dubliniensis or Tropicalis, but the combination didn't really work in things like Oris or Glabrata or Cruzii. So again, this means that these molecules are probably targeting a protein that is very specific in between these different species. And the way that we selected the best of them was using Candida albicans strains. So we could potentially go back to our initial library and select them again against some of these other species, and we would probably recover other molecules as being the best. But it was still quite encouraging that we were seeing some degree of effect in other Candida species. So do they potentiate other antifungals? So for other azoles, the answer was quite simple because we did see that they were able to potentiate other azoles. So we were doing the same type of assays by doing disc assays and then measuring the radius of inhibition with the drug combination. And what I'm showing here are these RAD values for the different combinations. So for example, you can see that the RAD values increased significantly relative to DMSO alone. These are, two different isolates. One of them being actually quite resistant. You see that it started at dark blue, so it means the RAD value was very, very small. And in many cases, the RAD value increased to orange, so it got very big. So this suggests that the drug combination was able to restore susceptibility in these assays. In contrast, we did not see synergy with the other antifungals. So with antifungals from the other classes, we really did not see much. So this was quite suggestive that indeed it must have something to do with the fact that we're inhibiting ergosterol synthesis, or we're somehow disrupting lipid homeostasis. So this prompted the question, well, will these molecules inhibit other inhibitors of ergosterol biosynthesis? And indeed, this is what we saw. So we used terbinafine and fenpropimorph, which are molecules that also target the ergosterol biosynthesis pathway, but either above or below fluconazole. And this heat map here shows relative susceptibility levels comparing these molecules against a negative control DMSO. So if it's white, it means there's no effect. If it's blue, it means susceptibility decreased. So it means we restored susceptibility. So the combination worked quite well. So these are 3 different isolates. Here we see treatment with fluconazole where we have synergy. But we also got a lot of blue, so a lot of effect with terbinafine and fenpropimorph. Again, this was dependent on how resistant the isolate was. So there's more blue with the more resistant isolates because there's a higher dynamic range. But it did suggest that indeed these molecules actually can synergize with other ergosterol inhibitors as well. And we also tested the sphingolipid biosynthesis pathway by combining myrisine with these molecules. And again, here we saw restoration of susceptibility with this combination. So suggesting that we're somehow disrupting lipid homeostasis, and potentially this is why we're synergizing with these lipid synthesis inhibitors. So to further probe this, we used a lipid droplet stain. So it's called BODP, and it basically stains kind of all lipid droplets in the cell. So it's a very descriptive marker, but you can see immediately if there's something wrong with this process. So for example, these are Candida albicans cells treated with DMSO, so the negative control, and those treated with fluconazole actually do not show any difference in this stain. But we were surprised to see that treatment of combination plus potentiators actually changed the patterns of staining for this dye. So we were now getting more lipid droplets and brighter lipid droplets. So there was we were disrupting something regarding to where the lipids went, where they accumulated, and maybe they're transported in the cell. But again, this is very descriptive and it doesn't tell us that much about mechanism of action. Because drug resistance mechanisms are associated with disruption of efflux pumps or of azole transport in the cell, we were also curious to see whether these molecules actually restored susceptibility by somehow altering drug transport in the cell. So the idea is that if you block efflux pumps, then you have more of the fluconazole in the cell, and then the cells would become more susceptible. So we thought, well, maybe these molecules could do something to drug transporters. So to ask this question, we used rhodamine 6G dye, which is a fluorescent transporter, a fluorescent substrate for ABC drug transporters for things like CDR1, CDR2. And the idea was, well, maybe in combination, we'll see more of this dye in the cell if it acts on drug transporters. So we did these combinations. We put our cells in a flow cytometer, and then we measured how much rhodamine 6G the cells had inside. And we actually did not see much of a difference with or without the small molecules, suggesting that blocking drug efflux is not the main mechanism by which these molecules act. So you'll see that this heat map shows the effect of the potentiators. And for the most part, we're getting white values, so things that are that mean no change relative to the MSO control. So, but we were actually quite intrigued to see, well, what happens if we take some drug transporter mutants and test them again in combination? And here I'm showing different ABC transporters because we tested rhodamine 6G, which acts on ABC transporters, as well as TAC1, which is a transcription factor that regulates CDR1 and CDR2 expression. And we wanted to see, well, will cells lacking these transporters or this transcription factor respond differently to the drug combination? And actually, we found that mutant of TAC1, which regulates expression of CDR1 and CDR2, was more susceptible to the combination. But in contrast, mutants of CDR1 were actually more resistant to the combination. So this was quite puzzling because if you are deleting this, this major fluconazole drug transporter, and that means you have more fluconazole in the cell, in theory, why would the combination actually make the cells more resistant to fluconazole? So this did not make any sense. But then we started reading a bit more about CDR1 and CDR2, and we did a few more experiments to try and understand what's going on. So what we did was to take CDR1 and CDR2 mutants and see, well, does fluconazole levels in the cell actually change in these mutants with these small molecules? And indeed, we saw that with CDR1, we were actually getting less fluconazole in the cell, which was surprising. So the potentiators appeared to decrease drug accumulation in the CDR1 mutant, which made no sense to us. But it makes a bit more sense when you think that CDR1 is actually not just a drug transporter, it actually participates in membrane biogenesis. So CDR1 is a phospholipid translocase. So it basically moves phospholipids from in to outside of the plasma membrane. It localizes to lipid rafts, and it actually actively participates in membrane biogenesis. So CDR1 is not just a drug transporter, and also both sphingolipid synthesis and ergosterol synthesis are necessary for the proper localization of this transporter to the plasma membrane. So in theory, if you disrupt any of these processes, which we think our molecules might disrupt, you get altered CDR1 localization and possibly function. So we think there's more going on in these cells with CDR1 other than just drug efflux. So this was another clue that potentially the combination is altering somehow membrane integrity and membrane synthesis that might lead to these weird effects with these drug transporters. So this is something that we're still trying to understand in more detail to see what's actually going on. So what about effects on virulence, or what about the toxicity of these molecules, or what's their impact on treatment outcomes? So it was quite interesting for us to see right from the beginning that these molecules did not have any intrinsic activity on their own. So when you take cells, incubate them with these molecules at very high concentrations, of either 30 or 37 degrees Celsius, we did not see any impact on growth. So this is relative to a DMSO negative control. But what we did see effects on was filamentation. So these are pictures of Candida albicans cells grown at 37 degrees for 2 hours, where we would get induction of filamentation. So with DMSO, we see the formation of these very nice and long hyphae. With fluconazole, we see a bit of an effect of decreased filamentation, but with the potentiators alone, we actually saw that these long hyphae were no longer forming. So we were getting mostly pseudohyphae or these very short stunted hyphae. And these effects were significant and apparent in different strains with different molecules. And the combination of fluconazole plus potentiator did not add like an extra effect. So it seemed like on their own, these potentiators did something to filamentation, possibly linked with their activity on lipid homeostasis, because we know we need to have proper membrane integrity and proper membrane synthesis in order to make these long filaments. So this was quite interesting to us, because it could affect these molecules could impact the virulence of Candida on their own. So we also tested their effects in combination with human cells. So we used PBMCs as well as red blood cells. And we incubated these human cells with different concentrations of molecules and asked to see whether they would lyse these cells, or they would be cytotoxic to these cells. And actually, we saw very little toxicity to these blood cells at concentrations where these molecules are active in combination with fluconazole. We also tested them in combination with fluconazole, and that did not further increase their toxicity. So we think that this is quite hopeful as a starting point for further development of these molecules. So how about impact on treatment outcomes? So to answer this, we used the Galeria melanella model. So this is a model quite broadly used for fungal infections because it can reproduce several features of fungal infections. So you can grow them at 37 degrees, and the Candida cells will filament and pierce through these worms. It's quite disgusting, but it can be quite a nice model to screen for antifungal molecules. So what we did here, we incubated first Galeria melanella with the molecules alone, so no Candida, and first asked to see, well, are these molecules toxic to the Galeria? Not that the Galeria would be humans, but it was still a good question to ask. So fluconazole does not have this toxicity effect, and we saw that our molecules also did not do anything to the Galeria relative to a PBS injection control. So how about when we add Candida albicans? So when we add Candida albicans, you can see that actually a lot of the worms die. So the blue line here is our worms infected with Candida and treated with PBS. When we treat them with fluconazole, we see quite a lot of restoration of survival. But it was interesting to see that when we treat these larvae with potentiators alone, we see a bit of survival and restoration. So we thought that this might actually have something to do with the fact that the molecules on their own inhibit filamentation. But what was actually nice to see was that the combination of fluconazole plus potentiators was able to lead to a significant change in survival relative to fluconazole alone. So both of these two dark lines show the drug combination, and you can see that their survival really increased relative to fluconazole alone. So to kind of further solidify this result, we basically took these larvae and tried to determine, well, how many Candida cells are in them. And we saw that indeed, relative to the PBS control, which had a lot of Candida inside them, the drug combination actually significantly decreased fungal burdens in the larvae. So you can look at these two two bars, the dark red one and the dark green one, and you can see that these are lower than the PBS and lower than the fluconazole treated groups by 10-fold or 100-fold. So we think that the increased survival was due to decreased fungal burdens in these larvae. On their own, the potentiators did not actually alter fungal burdens that much. This one here is significant, but we don't think this is a big change relative to PBS alone. And what we also did was to take these larvae and see, well, can we see the Candida inside? Are they still filamenting? And we saw that indeed with, when we treated these larvae with compounds alone, we were no longer seeing the long filaments that we were seeing with PBS treated larvae, suggesting that it's likely that we were seeing an effect on filamentation in these hosts, which could explain why the larvae were surviving slightly better when treated with the compounds alone. So to kind of leave you on a positive note, we're still working on this, and we have developed a second generation of molecules by doing multiple rounds of SARs, a structure-activity relationship, where we're taking the initial molecules and modifying their moieties and then trying to see, well, are these molecules better or not? So the idea is to basically get optimized molecules, and we're also still working on identifying their mode of action. So stay tuned. So with that, I'll just end this talk, and I want to acknowledge people that did most of the work on this project in particular. Cecile, Jan, and Paylene, as well as our collaborator, Jason Sello and Philip Alabi, who was a chemist in his lab that actually developed and generated these molecules. And also thank our collaborators and our funding sources for all of their support. And thanks for listening, and we'll be happy to take questions.